So I have the pleasure of introducing our speaker, Dr. Lindsay Burrage. Lindsay obtained her MD PhD from Case Western Reserve University and did her residency in pediatrics there, followed by a residency in medical genetics and a fellowship in biochemical genetics at Baylor College of Medicine. She's currently an associate professor with tenure, as well as the associate director for the medical genetics residency program at Baylor College of Medicine. She has authored over 110 peer-reviewed scientific publications and has a leadership role in the Center for Precision Medicine Models that aims to help researchers and clinicians to establish and study animal models of human disease-associated genetic variation. We'll hear her perspective as a physician scientist, leveraging the capabilities of a research lab to characterize the functional consequences of genetic variants and skeletal disease in this case and their mechanisms. Lastly, I have the pleasure of counting Lindsay as a close colleague for many years now and a good friend. I've learned a ton from her over the years, and I really hope that you can also get to learn from her over the next hour and a half. So, Lindsay, the floor is yours.
Thank you, Carlos. And thank you for this invitation to talk to you today a little bit about our work on precision medicine models for undiagnosed and rare diseases, in particular skeletal disorders, of which we'll talk about a few today. There we go. So the objectives of my talk today are to introduce the Baylor Center for Precision Medicine Models and discuss how the center can provide data that will impact clinical care of individuals with rare and undiagnosed skeletal diseases. We'll discuss strategies used to model human variants in mouse and discuss the potential utility of precision animal models as preclinical models for therapies for skeletal disorders. So rare and Mendelian disorders affect over 7,000 there are over 7,000 rare diseases that affect 25 to 30 million people in the US alone. And over 60% of those with rare disease are children. And many of our patients see many physicians before consulting an expert. And then this can take many years to occur. And even when the most comprehensive of genetic testing is performed, still about 60% remain undiagnosed. And even when a diagnosis is achieved, only about 10% of diagnoses are associated with an FDA-approved treatment. So over 90% of our rare diseases don't have a treatment. And so given this, the challenges of this diagnostic odyssey and knowledge gaps with regards to treatment in rare and undiagnosed diseases, we feel that animal models are just one strategy that can be used to address these gaps. And so animal models can help us because they can help us validate genotype-phenotype association. They can help identify biomarkers of disease progression. They can help us understand molecular mechanisms of disease pathology. And they can be important tools for assessing new therapies for a rare disorder. And so in response to this, in 2020, Dr. Jason Heaney, Dr. Alex Milosevic, and myself established the Baylor College of Medicine Center for Precision Medicine Models. And our goal was to support physicians, genetic counselors, patient family groups, and individual researchers in the development of precision animal models to end this diagnostic odyssey for patients with undiagnosed rare Mendelian diseases, but also to develop models that can serve as resources for researchers pursuing preclinical studies to investigate personalized medicine approaches to their care. And this center was established in 2020 with NIH funding. We're now in our second cycle, and our leadership team has expanded to include Dr. Shinya Yamamoto as well. Now, there are many different animal models that one can use for modeling rare disease. Our center focuses on two, the fly and the mouse. And the way our center works is that we accept variant nominations from the community. So anyone, a physician, a genetic counselor, a family group, a researcher can nominate a variant of interest for modeling within the center. I'm showing you our webpage with our nomination, link to our nomination form. We ask for information about the variant and basic HPO terms about the phenotype so that we can get an understanding if the variant can be modeled within our center. We also include some variant nomination guidance on this page, as well as a link for tracking the nomination from start to finish. So how do we decide which variants can be accepted into our center? Well, for each variant, we review both the bioinformatics of the variant as well as the phenotype very carefully. And one of the goals of our NIH funding is that our variant models must have a clinical impact. They must impact patient care in some way. So we take that into account. Our animal modeling teams review the animal modeling potential for the particular variant, keeping in mind the clinical question. And then our team meets together to review the variant and discuss whether or not we can accept a variant for modeling in our center. And when we do accept a variant for modeling in the center, we notify the submitter, and they become a partner from start to finish. They're a collaborator, they're involved in the design, the phenotyping, and we send the information back. And in some cases where there's a researcher who nominates the variant, we'll send the model to them for them to do the studies that they propose. And so the priority variants for the center are variants in putative novel disease-causing genes, variants of uncertain significance in known disease-causing genes with phenotypic expansion. But we'll also consider variants that are known to be pathogenic or variants of uncertain significance that overlap with the phenotype if the model can help us in evaluating biomarkers for diagnosis or monitoring of the condition, or if the model will then be used for developing therapeutics for the condition. So most of my talk today will focus on mouse because for many of our skeletal disorders, mouse is a better model than the fly if we're trying to mimic the phenotype of the patient. But for a host of other disorders, fly modeling certainly has its advantages. It can be quicker, it can be cheaper. We can leverage the capabilities of the Undiagnosed Diseases Network Model Organism Screening Center here at Baylor. It can give us a rapid characterization of the functional consequences of human variants and help us probe molecular mechanisms, which is really important. And I've included three examples of some of the published work from the fly. But as I alluded to, today I'm going to focus on some examples of where mouse modeling has helped us resolve a variant of uncertain significance and a gene of uncertain significance. There's a review that was published in 2020 by Burrage et al. That demonstrated that mouse models of skeletal disorders, over 90% of them actually mimic the human phenotype. And so mouse can be a great tool that we turn to for modeling various skeletal disorders. And so for mouse modeling, we tend to turn to the mouse when the variant impacts systems like the skeletal system or genes in which the fly is just not appropriate. We also use the mouse for validating human disease phenotypes if we really are trying to mimic the human disease phenotype for some of our phenotypes that we receive. And it's especially helpful for studying nutritional interventions and new therapies for the condition that we're modeling. And so what kind of models do we make? Well, in the simplest of terms, and in some cases we can do this, we can simply make a knockout or a conditional knockout mouse. We typically, in this case, will delete a single exon, which leads to frame shift and nonsense-mediated decay. But most of our examples, we can't get away with this easier approach. Many of the variants that we receive in the center are de novo heterozygous variants, and they require a knock-in allele. And in some of those cases, we can make a constitutive knock-in allele with genome editing, but especially for some of the early onset childhood phenotypes that are nominated with variants to the center, this can be problematic because if they're heterozygous and de novo, we can maybe introduce the allele, but we can't propagate it because the mouse won't survive. And so we have to come up with other strategies for modeling these heterozygous de novo variants that cause severe phenotypes, especially in early childhood. And so we have several strategies, of which I'm showing one for generating conditional knock-in alleles in this case. And we also have some other strategies I'll talk about on the next slide for doing some screens that can sometimes help in this scenario. In addition, we're increasingly getting requests to generate humanized alleles. In this case, we'll introduce a variant of interest within the human exon and replace one or more human exons with the variant. But in that case, we also need a referenced allele that's the wild type allele that's humanized as well for a control. So in that case, we would be generating two mice, one with the referenced humanized allele and one with the variant humanized allele. So this is just a taste of some of the strategies that we use. And then as I alluded to before, we are increasingly using screening techniques to screen for some of these variants. And so I have an example of that in the talk today. Traditionally, we would use a founder screen, although that can get somewhat complex. And so we started using a prime editing screen. And in this case, we take advantage of the fact that prime editing is not as efficient as traditional CRISPR gene editing is. And it allows us, in some cases, to generate low-level mosaic founders that harbor our variant of interest, but are still alive and viable. And then we can use these founders to then either through natural mating or sperm cryopreservation, generate heterozygous embryos that we can phenotype and look for the phenotype of interest, usually with embryo phenotyping, because we, in most of these cases, would expect them to not survive. And I'll show you an example of how this works in one of our examples today. So I told you what we do with variants that are accepted in the center, but what about variants that are not accepted in the center for modeling? So we are usually able to provide strategies for alternative ways of resolving the variant of uncertain significance. And we did that in over 80% of our cases. In some examples, this might mean referral to other researchers who we know have an interest in the gene, or that we've matched using a tool developed by Dr. Yamamoto here at Baylor called Model Matcher, which matches folks who have an interest in a gene from a research perspective with those who are looking, maybe because they have a variant of interest, they're looking to match with a researcher studying that gene. We've also been able to do some deep dives into literature, and in some cases our literature search has provided new data that helped resolve the variant, and the publication that I showed here is one example of that. And then we also can do matchmaking with our Baylor genetics database, and we've had some success where simply by finding additional cases, we were able to resolve the variant and no modeling was necessary. And then, although I talked to you about mouse and fly as our primary model systems, we are increasingly using cell models as well. We have, in several cases, used cell models where we've been able to do RNA sequencing, and that has helped us resolve a variant of uncertain significance, or at least helped us to define the effect of a variant before we tackled it in the mouse. And so we do have several examples of cases that we're able to resolve even without modeling, and it prevents us from generating models that may not be necessary. And so today, I'll tell you about two stories. These are two of our earlier cases, both of which came from the Undiagnosed Diseases Network. And I think both highlight different strategies that we use for modeling and getting answers back to patients. This first story, I'll tell you about biallelic variants in a gene called KMO and a novel congenital NAD deficiency. This work was led by a graduate student in the lab, Natalie Aceves-Ewing, in collaboration with Dr. Christensen and Dr. Su, who helped assist with our embryo phenotyping that I'll show you today. And so NAD, or nicotinamide adenine dinucleotide, is a coenzyme involved in many different biological reactions. We've all learned about NAD in biochemistry, and if you do a PubMed search, you'll come across thousands and thousands of articles about NAD. NAD is associated with many different processes: energy metabolism, mitochondrial function, RNA regulation, DNA repair, genomic stability, and protein modification, just to name a few. Back in 2017, Dr. Sally Dunwoody's group described a group of conditions called congenital NAD deficiency disorders that are associated with congenital anomalies. These anomalies included vertebral anomalies like hemivertebrae or vertebral fusions, limb anomalies, in some cases short stature, short limbs, as well as missing digits, a variety of different cardiac anomalies, renal anomalies, and as I mentioned, a subset of patients had short stature and a subset had global developmental delays as well. These congenital NAD deficiency disorders all involved enzymes of the kynurenine pathway, which is the pathway from which tryptophan is converted to NAD. Now, in addition to this pathway, which comes from, as I mentioned, tryptophan, you can also generate NAD from niacin in the diet through the Price-Handler pathway, as well as recycle it with the NAD salvage pathway. And the original, the first few congenital NAD deficiency disorders all involved enzymes in this pathway, KYNU, HAAO, and NADSIN1. And the variants in the gene that I'll talk about today is KMO. It stands for kynurenine monooxygenase, and it's the most upstream of the enzymes associated with congenital NAD deficiency disorders that have been described so far. So how did we get interested in KMO? Well, two siblings, two girls, were nominated or applied for admission into our Undiagnosed Diseases Network site at Baylor. One of them was deceased, the other was alive. Both had congenital anomalies. These included vertebral and rib anomalies, as well as a variety of cardiac, pulmonary, renal, and inner ear malformations. And the living child is now much older, and she has short stature as well. The living child had comprehensive genetic testing at the time, which included trio and even quad exome sequencing, and no, it was not, both were non-diagnostic for both children, but a research analysis identified biallelic variants in KMO as the leading candidate. I'm showing the variants here on the pedigree. One of the variants impacts a -1 canonical splice site inherited from the father. The other is an amino acid change inherited from the mother. And both affected siblings had both variants, whereas the unaffected siblings, one was a carrier for one of the variants and the other did not inherit either variant. Now, given the nature of the variants, we hypothesized that these were likely causing a loss of function of the KMO enzyme. And as such, we hypothesized that if KMO was deficient, that the most upstream metabolite, kynurenine, should increase. And to test this, we collaborated with Dr. Dunwoody's group, and targeted metabolomics was performed, which revealed an increase in kynurenine, 6.6-fold increase. In addition, her team was able to show a reduction in NAD as well, which would be expected and consistent with our hypothesis. We were also able to send plasma and urine on the living patient to Baylor Genetics for clinical metabolomics analysis, which confirmed this as well. And so, knowing that we likely had a loss of function, we decided to take kind of the easier approach for modeling. So rather than introduce each of the variants, we decided that a knockout mouse would probably be sufficient for this project to determine whether or not a loss of a reduction of this enzyme activity increased risk for congenital anomalies. And so I'm showing the design here. We did a single exon deletion, which led to a premature stop and a frame shift premature stop, and was predicted to undergo nonsense-mediated decay. And Natalie was able to validate this allele in a number of ways. Here I'm showing the Western blot of the liver, where you can see absence of the band for KMO in the knockout animals compared to their heterozygous or wild-type litter mates. In addition, we validated biochemically as well. So just like in our patient, we expected an elevation in kynurenine in the serum of these mice, and we were able to confirm that. And then what I haven't told you so far, though, is that we were a bit puzzled because the KMO knockout mice were viable and they were even fertile. So they didn't look like our patients who had significant congenital anomalies. But we hypothesized that this was because the mouse diets are chock full of niacin. And so we hypothesized that if we remove the niacin from the diet, these mice would not fare well. And sure enough, Natalie was able to demonstrate that the KMO mice, which are shown with the red lines here, lose weight when they're placed on a niacin-free diet, whereas the wild-type mice continue to thrive because they're able to, presumably because they're able to generate NAD from tryptophan. So I already alluded to some of the fundamental differences between mouse and human NAD metabolism. First, the mouse breeder diets and the chows are really rich in NAD precursors, and we had to keep that in mind when designing our experiments. Moreover, humans rely more on niacin than tryptophan for NAD synthesis, and that's believed to be because humans divert some of the kynurenine metabolites to the citric acid cycle, and thus rely more on niacin from the diet to generate NAD. And so we kept this in mind when we designed our experiments, and we also used the model that Dr. Dunwoody had previously shown with some of her mouse models, where we crossed a KMO dam with a heterozygous KMO, so a KMO knockout dam with a heterozygous KMO male. And we did this so that we could mimic the niacin dependence that's typical of humans and generate both control mice as well as knockout mice for our studies. So these mice were crossed, and once we noticed a plug, the dams were placed on either a control diet or a low niacin diet. And then at E18.5, the embryos were evaluated through visual inspection as well as micro CT in order to see if the embryos that were from the dams on the low niacin diet were at higher risk for congenital anomalies. And so here I'm going to show the percentage of congenital anomalies that were noted based on genotype. First, this is the control diet, and you can see there was no significant difference between the percentage of embryos with one or more soft tissue anomalies between genotypes. However, when we did the same for the low niacin diet, we noticed several embryos had external anomalies that were visible on exam. These included anomalies of the tail, anomalies of the limb. You can see some retrognathia on this mouse as well. But we got an even clearer picture when we did micro CT of these embryos, and I'm going to show some of those examples here. So here's, you can first see some of the external anomalies like the curled tail, but when you get an internal look, you can see cleft palate. We saw heart defects in several of the mice. Here's an example of missing kidneys. For this particular group, you can also get an idea of the skeletal anomalies. We saw several different vertebral fusions. I'll show some examples here. Here's a C5, C6 fusion, as well as a C3 and C4 fusion. And then we also, in some cases, saw fusions in the thoracic area. Here's a T5, T6 fusion. And so when we summarized all of this data, we found that the KMO knockout embryos who were born or who were from dams fed a low niacin diet, they had a higher percentage of embryos with one or more soft tissue anomalies compared to the heterozygous embryos. And so this was consistent with our hypothesis that the genotype together with the low niacin diet increased risk for anomalies. Now, we wanted to do one last thing to confirm that our hypothesis was correct. We wanted to confirm that the embryos from the low niacin diet, which were KMO knockout, had a lower NAD level. And so here is the data showing the NAD levels from knockout and control embryos on the control diet, and NAD levels not significantly different. And here is from the low niacin diet, and as we expected, the KMO knockout embryos have lower NAD, significantly lower NAD than the control embryos. And so with this first example, we hope to have convinced you that KMO knockout embryos are at a higher risk for congenital anomalies, including skeletal anomalies, in the setting of a low niacin diet, and that KMO deficiency is a novel form of congenital NAD deficiency and a risk factor for congenital anomalies in humans. In addition, I think this example highlights how NAD has a critical role during embryonic development, although the precise mechanisms underlying this remain unclear and further work needs to be done. And so here I'll shift gears to talk about a second story where we used a different modeling strategy to demonstrate that a novel axin 2 variant disrupts Wnt signaling and has a unique phenotype highlighting the role of axin 2 in development and disease. This work was also led by the same talented graduate student, Natalia Savas-Ewing. And this work we had collaboration from Drs. Denise Lanza and Dr. Logan Su. And I should mention Denise, as well as Jason Heaney mentioned on this slide, both helped with the mouse modeling for both of these examples and lead our mouse modeling team. So how did we get interested in axin 2? This was also one of our very early submissions to the Center from the Undiagnosed Diseases Network here at Baylor. And this variant in axin 2 was identified in a child who had oligodontia, sparse hair and eyebrows, absent or underdeveloped nails, and hand and foot skeletal anomalies. I'm showing you pictures of her feet here. She also had a short palate and significant feeding issues as a result early in development, required a prolonged NICU stay. She had exome and then genome sequencing, and both identified the same variant of uncertain significance, a de novo heterozygous variant in axin 2. Now, axin 2 is a known disease gene. Variants in axin 2 are associated with a condition called oligodontia colorectal cancer syndrome. And this particular condition is associated with oligodontia, sparse hair and eyebrows, and colorectal polyps, as well as colorectal cancer. And our patient had some of the phenotypes that overlapped. And one hypothesis was that maybe she had two diagnoses. Maybe she had this condition and a second variant that was driving her other anomalies. But an alternative hypothesis was that this was a phenotypic expansion and that this novel axin 2 variant is actually causing a more severe phenotype in this patient. So in order to kind of start to tackle that, we looked at the variants that had been previously associated with oligodontia colorectal cancer syndrome. And you can see many of them are frame shifts or early stops that are scattered throughout the gene, shown in black, and a handful of non-synonymous variants have been associated as well. But what's different about the variant in our patient is that it's an amino acid change in the tankerase binding domain. And to our knowledge, there hadn't been a variant before associated with oligodontia colorectal cancer syndrome that was located in this particular domain. So this made us think maybe this variant is acting a bit different than these others. And so what does axin 2 do? Axin 2 is a scaffold protein with a complex role in canonical Wnt signaling. It can be a negative regulator with the beta-catenin destruction complex, but it can also be a positive regulator through LRP5/6 Wnt receptors. And the tankerase binding domain is important because tankerase binds axin 2 and parcelates it, and that's one way it regulates the levels of axin 2. And given that our amino acid change specifically impacted the tankerase domain, we hypothesized that it somehow impacted this interaction. Now, this is a heterozygous de novo variant. And as I alluded to before, these can be particularly problematic because, the child who had this variant had a lot of feeding issues. She was in the NICU for a long time. And as a result, we hypothesized that the heterozygous mice, if we could make them, they probably wouldn't survive because they would potentially have feeding issues as well. And thus, if we generated a mouse, we wouldn't be able to propagate the line. And so we decided we could either take a conditional knock-in approach, which would take some time to generate, or we could try one of our screens. And as I mentioned, traditional CRISPR-Cas9 gene editing can be very efficient, and it can cause lethality, especially if the variant were introduced on both copies. And it can also be somewhat challenging to tease apart if multiple variants are introduced, et cetera. So we decided to approach this with a prime editing approach to take advantage of its lower efficiency and higher accuracy for point mutation. So how do we do this? Well, we did prime editing and generated F0, so founders, and we genotyped them and Sanger sequenced them. And fortunately, we were able to identify a mosaic male founder that was about 20%, had 20% mosaicism for the variant of interest. And it fortunately was viable. And so we were able to do an N1 screen of its offspring. And so we first did this with live births and looked to see with our live births that were born to this male, what their genotype distribution was. And when we did that, we found that all of them were wild type, none of them were heterozygous. And so this could mean that our editing just didn't work, or it couldn't be passed on, or perhaps it meant that it was associated with embryonic lethality. So to test this, we collected E18.5 embryos and genotyped them. And sure enough, we got about a 50-50 ratio, which is what we'd expect. So this suggested that these embryos were present at E18.5 right before delivery, but were not surviving the postnatal period. So the next thing we did was work with our embryo phenotyping team and do both external and internal evaluation. And the external evaluation revealed some limb anomalies, some missing digits, and some curled tails. And you can also see that a few of our mutant animals, our embryos, were smaller than the others. But none of these seem to explain why the mice weren't surviving. Curled tail and a missing digit wouldn't impact survival. So we also took a closer look internally through micro CT. And so here I'm showing you a picture to get a closer look of the craniofacial area. And what we noted in all of our heterozygous mutant embryos was what appeared to be a shortened palate. You can see the palate does not extend all the way back to the epiglottis like it does in the wild type animal. And we first thought this was a short palate, but it turns out when we did a closer look, it's really a soft palate cleft. And we saw this in all of the embryos, the heterozygous mutant embryos. Here's a closer look on this slide. And we suspect that this is what's impacting survival because they're potentially regurgitating, it's impacting feeding, and they're not able to survive. And so I'm not going to tell you I'm not going to have time to tell you the entire story, but overall, we felt that overlapping phenotypic data from the humans and from the mice. And we also had some fly data as well, demonstrating and supported that this heterozygous E66K variant in axon 2 is pathogenic. One of the things we noticed in looking at the anomalies that we did see was that the kinked tail is something that's typically associated with increased Wnt signaling in mouse, and the soft palate cleft has been associated with decreased Wnt signaling. And so we do think that the effect of this variant may be context dependent, and the fly work also supported that. Lastly, as we were pursuing the mouse studies, we were trying to find more patients that had similar variants in order to provide further support that our hypothesis that this was a phenotypic expansion was correct. And but we knew that there are a lot of variants in axon 2, and so we wanted to be smart about how we started to look for other variants that might be associated with an overlapping phenotype. And so to do that, not only did conservation analysis with using both axin 1 and axin 2 in humans and mice, and then she also did a search of NOMAD, which is a database of relatively healthy individuals, and she plotted the number of variants at each position of this tankerase binding domain of axin 2, and she plotted the number of variants in NOMAD that she saw, and our hypothesis would be that if the variant was causing this severe phenotype, like in our patient, we wouldn't see any variation in NOMAD at that position. And sure enough, our position at position 66, where our patient's variant lies, there was no variation in NOMAD. And likewise, position 65 and 67, both are conserved in axin 1 and axin 2, mouse and human, and both also, like the position 66, have no variation in NOMAD. So we hypothesize that at least variants at position 65, 66, and 67 would produce this, an overlapping phenotype. And the same could be true with other positions, but we decided to focus specifically on these two neighboring positions. And through GeneMatcher and largely ClinVar searches, we were able to identify patients with overlapping phenotypes who had variants at position 65, 67, and even another patient with a different variant at position 66 that overlapped as well. And so that's the finding of additional cases helped with similar phenotypes, helped provide even further support that this was a phenotypic expansion. And these patients, as I mentioned, had overlapping phenotypes. There was one, though, with the E66K variant that was an adult who also had a diagnosis of colon cancer. So we think that that means these patients are at risk for colon cancer, just like the oligodontia colorectal cancer syndrome, but they also at risk for these other phenotypes as well. And so the conclusions for the second part is that novel variants in the tankerase binding domain, specifically at position 65, 66, and 67, expand the phenotype of axin 2 related disorders. And that this example shows the potential benefits of doing an N1 prime editing screen when you're trying to study potentially lethal de novo heterozygous variants in the mouse. And I didn't have a time to talk about the fly modeling, but the fly modeling together with the mouse modeling supported a context-dependent effect of the E66K variant. And if you want to read more, our preprint is up on medRxiv. So overall, with both of these stories, I hope that I'm able to highlight how precision models can play a critical part in validating novel disease gene discovery. These models have important roles in determining molecular mechanisms of disease-causing variants and helping us to resolve variants of uncertain significance. They're essential resources for preclinical studies of nutritional and medical interventions, and we welcome your variant submissions to our center as you encounter them in your routine clinical care of your patients. We have a lot of acknowledgments because this type of work is really a team approach. It takes a village. We want to thank all the collaborators for both the axin 2 as well as the KMO project, all of the members of our different cores, including the co-PIs in this grant, including Dr. Heaney, Dr. Milosevic, and now our newest MPI, Dr. Yamamoto, our funding sources, my own lab, and then I wanted to end by asking you if you have variants, and if so, we'd love to collaborate. Here's our QR code, and you can submit to our center, and we're happy to work with you. And with that, I'm happy to take any questions.